Hi, Dr. Woeller here for Integrative Medicine Academy. Um, Very much appreciate everybody joining me for this. What I really feel you'll find very, very interesting and informative lecture. We're going to talk about the gut microbiome, mucosal invasive candida, and we'll look at Clostridia bacteria as well. And I'm going to weave all of this together. I would really encourage you to hold on to the end because I'm going to be introducing some newer information that has come out about Clostridia bacteria that's really important. And I'm also going to share with you some other resources that we have through Integrative Medicine Academy. So when we think of the microbiome, right, we're obviously all the time thinking about gut microbiome. It's important to understand that we have a skin microbiome too. We have a microbiome in our mouth, but we're going to focus primarily on what's happening in the digestive system. And so the microbiome is really this collection of organisms that exist on our body, inside our body, and particularly within the digestive tract. And I actually like this description from this particular 1988 article, a characteristic microbial community occupying a reasonably well-defined habitat which has distinct physiochemical properties. So that's really important because the effect of the microbiome isn't just influencing what's happening in our digestive system, although it plays a huge role in that. It influences many other things throughout our body. And so this was kind of an interesting slide. So we know the microbiome helps protect against pathogens. It helps in the synthesis of vitamins and actually makes many nutrients more bioavailable for absorption. It plays a huge role in the immune system. In fact, there's entire disciplines within biology that are purely dedicated to mucosal immunology. One of the things I didn't really appreciate, and actually until I started digging into this a little bit more, is promotion of intestinal angiogenesis. So it actually helps with improving blood supply, the microbiome does. It promotes fat storage. And we might think that's a bad thing, but if you look at all of our organs, pick up any anatomy book, you'll see that most of our organs, our internal organs, particularly the omentum that overlays and is attached to our intestines, is fat. So it actually helps in preventing against internal organ injury. The microbiome plays a big role in short chain fatty acid production. So butyric acid, for example, is very important. Many of these short chain fatty acids are also used in metabolism and can be help support in mitochondrial function as well. And I've done a lot of lecturing over the years about the influence of the microbiome on the central nervous system and vice versa, particularly from a pathogenic standpoint, when we have pathogens like Clostridia bacteria that are known to produce various compounds that can cross the blood-brain barrier, but there's also a transport of certain microbial compounds that can find their way into the brain and nervous system through the vagus nerve. And that's well known in autism, for example. It's also known in Parkinson's disease. And so all of us should try to work at trying to maintain the health of our microbiome as well as our patients. And the thicker our microbiome is, or I should say, the thicker the mucin layer that is in part induced by the existence of a healthy microbiome, the more buffering we have against toxins that could be damaging the epithelial cells. Because whenever we are damaging epithelial cells, we have an increased potential for for presenting what's called antigens, which are compounds that can be, well, normally recognized by the immune system, but presented to the immune system. And if it's done so in a robust way, it can actually trigger inflammation. And so we want to talk about intestinal immune dysregulation, immune activation in general, and intestinal permeability. And there's a lot of things that can drive that. So it's not just one thing. It could be food sensitivities. It could be inflammation in general. It could be chemical toxins we're exposed to, chronic infections, drugs, alcohol, even mycotoxins. We know that and mold that can colonize in the digestive system can lead to leaky gut, right? And this is fairly well known. As we start to break down the tight junctions of the epithelial cells, we start absorbing antigens, these compounds that trigger immune reactions. Some of these could be proteins, some of these could be other aspects of food, some of these could be compounds of microbes or intact microbes, find their way through the filtering mechanism of the gut, get presented to the immune system, and then can trigger inflammation. And this is one of the things that can also trigger autoimmune reactions, where we start forming immune reactions to our own tissues because the molecular patterns of some of these antigens will look or can look very similar to molecular patterns within our body. That could be our joints, our muscles, aspects of our mitochondria, our brain, etc. And one of the major things that drives leaky gut or increased intestinal permeability is candida. So candida has a number of different pathogenicity mechanisms where it creates problems along the mucosal lining of the digestive tract. But one of them is growing a hypha, a root structure that will burrow itself between the tight junctions of the epithelial cells and therefore increasing the potential for antigen presentation. And you can test for intestinal permeability. This was just an old test that we used to do from a lab that no longer is in existence called Biohealth Laboratory. You can actually do intestinal permeability testing through Genova. I'm not sure if there's any other labs out there doing it, but it's an interesting test. It doesn't tell you exactly what's causing it. It's just telling you that you have a problem. Lactulose mannitol absorption at high rates indicate that that filtering mechanism, the tight junction spaces between the epithelial cells are compromised. And so this is just somebody that has severe leaky gut. Well, this gets important when we think about food reactions or other environmental chemicals that we're exposed to, and the mixing of these things that get mixed up within our digestive system. And then the coexistence of candida, which is a commensal organism in our digestive tract, or the infiltration into our digestive tract by other opportunistic pathogenic bacteria like Clostridia. All of this can lead to problems that occur within the gut, triggering of the immune system, irritation, inflammation, and destruction along the mucosal lining of the digestive tract. And that can then lead to chronic disease. It might just be inflammatory bowel disease, or it could lead to some other type of autoimmune problem. And this is profiled very nicely in a very in-depth article by Dr. Aristo Vojani from Immunosciences Laboratory called The Role of the Exposomes in the Pathophysiology of Autoimmune Disease. It's actually a two-part article. If you're ever interested in reading it, you can find it online for free. But I wanted to show this slide because I think it's important for all of us to remember and recognize that none of us live in a bubble, so that we're all exposed, and some of us are exposed to different things or more things that can compromise our health and certainly compromise our microbiome. And that when people have microbiome problems, it's not just a deficiency of, let's say, a particular bacteria, or it's not just usually occurring from one thing. It's multiple things that usually contribute to it. But all of those factors can end up compromising immune function that allows for candida, for example, to become problematic. And so let's take a little bit of a deeper dive into candida from gastrointestinal candidiasis to what's called candidemia. One of the things to keep in mind is that candida, we know, is in many of the species anyway, are a normal commensal organism, a part of our microbiome. We can't get away from that reality. Just it's everywhere in our environment, just like for most part, these other microbes are in the environment as well. But Candida albicans is the most common type of candida, and it's responsible for approximately 400,000 systemic infections annually with a very high morbidity rate or mortality rate, excuse me. When I was an intern right after medical school, I remember there was a young woman that I was on an internal medicine rotation. And I don't remember her exact age, but I do know that she was in her 30s, mid 30s, and she had bowel cancer. And so she had a resection surgery and she was undergoing chemotherapy. And she ended up developing an invasive candida infection, essentially candidemia. And they were treating her with intravenous amphotericin B, and she died. So she was so immune compromised. And I've heard over the years, in different types of medical channels that, people often minimize or diminish the existence of candida in the digestive tract as though somehow it's normal, so therefore it's not a problem. Well, it may be normal, but it can be a problem for many people. It turns out that the human digestive system is the largest reservoir or natural reservoir of candida in our body and is the major source of systemic fungal infections. And so again, there's many different types of candida, but candida albicans would be number one. So keep that in mind when you start thinking about patients who are dealing with chronic candidiasis. And we know that candidiasis is a fungal infection that's linked to any form of candida species. Now, it might just be on the skin. It might, you know, be occurring on the scalp, for example. Those aren't technically or typically life-threatening problems. People can develop oral thrush, commonly seen sometimes in young children or in elderly patients. And it can certainly be problematic, particularly for an elderly individual where they have so much candida irritating the mouth and the esophagus that they have a hard time swallowing. So oral thrush is certainly a real thing, esophageal candidiasis, and then of course cutaneous candidiasis. Now I would make the argument, at least particularly with oral thrush and esophageal candidiasis, is probably the candida isn't just there, it's probably all throughout the digestive system. And so gastrointestinal candidiasis is something that we encounter quite a bit as functional and integrative practitioners. In fact, in autism, for example, we know this is a major issue. It often contributes to digestive problems, but it can contribute to cognitive problems, behavioral issues, even speech and language problems with some of these kids. And one of the ways it is detected is through organic acid testing, particularly looking at high levels of arabinose, for example, as a reflective compound of the existence of invasive candida within the digestive system. So I have spoken before in many other lectures about different pathogenicity mechanisms of candida, and I wanted to show you this particular slide because this gives at least a bit of an overview of some of the ways candida can become problematic. So it can produce an enzyme called invasin that allows for its hypha to invade, but more specifically, through invasin, the candida can be endocytosed. So you can actually get candida that can get burrowed inside an epithelial cell almost intact. The hypha can grow through an epithelial cell. It can actually grow between the epithelial cells, between the tight junctions, for example. And what's interesting about that is it actually can translocate other organisms. So it can translocate other candida or even bacteria like staph or strep into some of the deeper tissue layers within the gut. And then if it went really severe, there certainly is the possibility for infiltration into the body to become systemic. Candida lysine is kind of a unique property of invasive candida. It produces this lysine compound that actually degrades and damages the epithelial cell through this lysine toxin. And this was relatively recently discovered. It's not something that, at least from my reading and my understanding, has been around for a long, long time. It's been around. It's just that it was relatively recently described. And one of the things that candida lysine does is it destroys the epithelial cell. and that can cause inflammation and then cellular damage. Now, before we would develop candidemia, we would have to go through, in most circumstances, mucosal invasive aspects of candida. And then, of course, we're getting compromised immune function along the way. So, for most of us in a functional integrative medicine practice, we're not going to be seeing people with candidemia. That was that patient that I had told you about in the hospital many, many years ago. But candidemia, this invasive aspect, this systemic infection, can really affect almost any organ system in the body. So we see here, for example, candida infiltrating into the spleen, and that would seriously compromise overall immune function. And again, there are many other mechanisms of candida pathogenicity: acetaldehyde toxicity, biofilm formation. Candida is known to produce gliotoxin. We often think of gliotoxin as mostly being linked to aspergillus mold. Well, it turns out candida can produce it as well. It can create immune dysregulation, mitochondrial dysfunction, pituitary adrenal axis activation. It's interesting, invading candida will induce the body to produce interleukin 6. And interleukin 6 will interact with the pituitary gland as well as the adrenal cortex to produce cortisol. And then, as I mentioned, translocation of other pathogens. So let's shift gears and just focus a little bit on some of the fundamentals of the immune response to candida. And we know when we think of our immune system, we have our physical barriers, our skin, our mucosal surfaces, for example. And then we have the innate and adaptive immunity. Now, the innate immune system is made up of things like monocytes, macrophages, neutrophils. And these cells don't typically need to be primed. Okay, they're going to react pretty much the same way each time. So they're not intrinsically affected by prior antigen exposure. Whereas something like the adaptive immune system adapts, changes, and shifts. It has to be trained over time. That's why typically with antibody production after an infection, it takes a good 5 to 7 days for those antibodies to show up. And so there is a communication that occurs between the innate and adaptive, and between the adaptive back to the innate immune system. And if we break this down between the individual cells, there are a few key players. On the right-hand side of the screen, from the neutrophils, eosinophils, all the way to the platelets and erythrocytes, this is linked more to what's called the myeloid progenitor cells. And so these are mostly the innate immune cells, for example, like the monocytes, mast cells, basophils. Erythrocyte isn't really considered an innate immune cell. And on the left, linked to the common lymphoid progenitor cells, B cells, T cells, and natural killer cells make up the adaptive immune system. Neutrophils play a huge role. They've circulated a very large amount, probably about 50 to 60% of all of the leukocytes that are produced. And they're in general circulation, and then they get called into a specific tissue area by different types of chemicals that are produced by the immune system. But they play a big role in the elimination of candida at the tissue level. So this is just an example of a candida organism, Candida albicans, being phagocytized by a neutrophil. So the neutrophil will come in, completely engulf it, and then the neutrophil has granules, so it forms something called a phagolysosome. It'll break down the candida and then essentially eliminate it. And so there is going to be some low-level inflammatory response second to this process, but if it's well regulated, well timed, it's not an issue. Our adaptive immune system is primarily made up of B cells and T cells. The B cells are what are producing antibodies. So that would be IgM, IgG, IgE, and IgA, for example. Our T cells are many different types: T helper 1, T helper 2, T regulatory cells, and then something called T17 cells. And so these help B cells in making antibodies, for example. They have a regulatory role. Some of them, like the cytotoxic T cells, can be directly involved in killing a cell that has been infected, let's say, for example, by a virus. So we have our innate immune cells on the right, we have our adaptive immune cells on the left, we have neutrophils, which play a major role with regards to microbial surveillance along the mucosal layers of the body. Candida, for example, it does so internally as well. And then there's one other cell I just wanted to mention called a dendritic cell. The acronym is DC. Notice the dendritic cell looks like a starfish. There's a reason for that. Is dendritic cells are strategically placed to sample fluid in fluid compartments to be able to sample a wide variety of different antigens that can be coming into the body. So for example, in our digestive system, we have dendritic cells that are reaching up, for example, and let me see if I can draw this. These dendritic cells with their little arms are reaching up and they're sort of situated in between certain epithelial cells. Okay. And so what they're doing as things are moving through the lumen of the digestive tract, these dendritic cells are sampling stuff, trying to recognize what is friendly and what is unfriendly. And when it comes in contact with some type of molecular pattern of an organism or some other compound that's not inherently intrinsic within our body, it will present or can present that information to adaptive immune cells that are sitting within our lymph system, primarily a lymph node that is linked to a specific region. So we have lymph nodes that are actually linked, for example, near our digestive system. And so those dendritic cells will take that information and go present it to a T cell. Okay, so this is a little bit of a better diagram. So we have an antigen. Okay, an antigen would be a compound of some sort that can elicit an immune response or a recognition response from the immune system. And that antigen gets taken up by an APC. APC is antigen presenting cell. So we have many different types of antigen presenting cells, but the main one, the most robust, what's sometimes considered to be the professional antigen presenting cell, is the dendritic cell. And that antigen presenting cell, that dendritic cell, will take that antigen and process it and deliver that message to a T cell. Now, if it were, let's say, a viral particle that has found its way into the cytoplasm of the dendritic cell, it's going to immediately send a signal into the immune system to a CD8 T cytotoxic T cell to just go in for the kill. So we're just going to get an immediate cellular mediated response to try to take out that cell. If it's more nuanced, meaning it needs to be processed in a different way, the antigen presenting cell will deliver that message to what's called a naive T cell. That naive T cell here is a Th0. And depending on what needs to happen, that message can then be sequenced either to become a Th1 cell, which might go off and form memory T cells, for example, or it might crosstalk back to the innate immune system and tell a macrophage, this is a macrophage, for example, right here, that it needs assistance. Or it might say, you know what, we need to make some antibodies to be involved in this process. And so it can, through different chemical signaling, it can trigger B cells to make plasma cells to make antibodies. So the antibody could be IgM, IgA, IgE, for example, depending on what the antigen was. And so it's important to recognize that these antigen presenting cells are connecting the innate immune system to our adaptive immune system. Now, before I go on, one of the things to remember about an antigen, okay, an antigen is a structure. It could be a protein, it could be a carbohydrate, it could be a phospholipid, it could be a nucleic acid, and they have different regions on them called epitopes. And an antibody will attach and recognize different epitopes. So an epitope is just a different type of molecular structure. And so there could be all different kinds of epitopes that might be linked to a given antigen. And so we can have different antibodies that then get produced to attach to a different epitope. So for example, candida, in order for our body to form an IgG against candida, there has to be some sort of recognition of an epitope on that candida organism to elicit some type of immune response. And where we get into trouble with regards to these chronic infections is that when a chronic infection like candida is recognizing, or the immune system is recognizing an aspect of candida, but the epitope of that organism, of a candida, for example, starts to interact and cross-react with our own mucosal tissue, or basically our own tissue throughout our body, that can start to elicit what's called a cross-reactive response. So cross-reactive types of antigens can trigger cross-reactive antibodies. And this is how candida, in part, can start to be involved in different types of autoimmune reactions. There's many mechanisms that are involved in this. Candida might just purely cause a leaky gut, increased intestinal permeability situation, where other antigens now pass through sort of unfiltered, but it can also start to destroy the lining of the digestive tract that also elicits an immune response. Add on top of that the fact that all of us consume food, and in our food are different molecular patterns of the food that we're eating. If those things start to elicit an immune response, now we have this entire mixture occurring. And that's essentially kind of this concept of what's called the exposome, potentially, is this collective things that we're exposed to that get mixed up and start to present themselves to the immune system, that then triggers these broader immune reactions. And so this is really sort of the beginning aspects of certain autoimmune types of problems. Now, one of the interesting things about candida is its ability to evade host immune recognition. And this gets fairly complicated, but these organisms have been around for, millions of years essentially. And so they've learned to adapt to our body, to animals' bodies, to other organisms, and to survive. And I always look at it this way. We as human beings have an intrinsic desire to thrive and survive. Well, the same thing occurs with animals, microbes, and particularly in things like candida. They have their own intrinsic desire to thrive and survive. And so candida, through various mechanisms, can actually inhibit macrophage infiltration. It can block the body's ability from recognizing and invading hypha. It can also shield itself in part from receptor recognition of our immune system. A specific layer that occurs within the cell wall of candida is called beta glucan. Now, there's glycoproteins, there's something called chitin, all of that makes up the cell wall. But there's something about the beta 1,3 glucan that it tries to hide and tries to shield. And it primarily has to do the fact that the beta glucan is highly immunogenic, meaning it's highly inducing of immune reactions. Well, certain bacteria, like gram-negative bacteria, have lipopolysaccharide as part of its cell membrane. Other organs have mannins or peptidoglycans or whatever these things are, right? These different chemicals. Well, they all have a molecular pattern. So every organism has a molecular pattern. And if it's an organism that typically doesn't reside in our body, it's essentially, when it comes into our body, would be considered foreign. But candida can live in our body, but it's also opportunistic. And so it contains or manifests different molecular patterns as part of its cell membrane, but the beta glucan is highly immunoreactive, so it tries to shield it and hide it so that our immune system doesn't recognize it easily as a problem. So every pathogen has a pathogen associated molecular pattern. The way to think of it is it's just every organism, every pathogen, for example, has its own unique molecular pattern that makes up its cell membrane. In our body, we have something called pattern recognition receptors, and these are proteins that are expressed on our cells that recognize certain types of pattern or pathogen associated molecular patterns. And these pattern recognition receptors, which are intrinsic to our body, are profoundly robust on things like dendritic cells, macrophages, neutrophils, epithelial cells. So our innate immune system has this innate ability to recognize certain types of molecular patterns through these pattern recognition receptors. Now, as you can imagine, there are many different types of pattern recognition receptors that have evolved over time to recognize these other opportunistic organisms that live in our world and sometimes find their way into our body. Now, these pattern recognition receptors are I won't go into it in great detail, but there's a limited amount of these receptors that have developed over time. And so our innate immune system does a really good job at stopping invading organisms early on. And if it's healthy enough and robust enough, it can pretty much take care of an invading infection pretty well. But if that infection has figured out a way or somehow the innate immune system has become compromised, then we need additional help. And that's where those dendritic cells, those other antigen presenting cells, will present aspects of the invading organism to the adaptive immune system. So we get a much more broad range of immune activity. And that would be things like our antibody production. So you don't have to remember all of this, but just to understand that these different pattern recognition receptors are toll-like receptors, are dectin-1, are dectin-2 type receptors, have been created and developed over time to recognize different molecular patterns of the cell wall of different pathogens. So there's different molecular patterns of candida that are being recognized by different pathogen receptor uh, uh, pattern recognition receptors. I mentioned beta glucan. And beta glucan, this 1,3 beta glucan, right here, gets recognized by dectin-1. And all of these pattern recognition receptors, whether it's a dectin or a toll-like receptor, whatever, will sequence downstream through different signal transduction pathways to induce the genome of the cell to induce different chemicals, different proteins. So it might be a pro-inflammatory cytokine, it might be different interleukins that are then directing activity either directly against the pathogen or now inducing activity of the innate immune system. Okay, so that is really key and fundamental. I'm only going to show you just an example because I'm going to come back to this MyD88. Okay, so MyD88 is a type of signal transduction pathway internal within the cell that gets triggered by certain types of pattern recognition receptors. So toll-like receptor 2, for example, or toll-like receptor 4, for example, are connected to that MyD88. Okay, well, that's not to say that MyD88 is only linked to candida. There can be other bacterial, for example, that might get recognized by the pattern recognition receptor that's actually connected to that pathway. The point I'm trying to make is that one of the major things that gets activated at the cellular level is a specific transcription factor called NF-kappa B. And NF-kappa B gets activated by a pattern recognition receptor of some type of pathogen, candida, for example, and it tells the genome of that cell to produce certain types of chemicals. So, for example, if we look at this particular slide on the left, we can see we have all of these different types of pattern recognition receptors that can recognize, some of them anyway, different aspects of candida. It'll recognize a certain PAMP, a pattern associated molecular pattern like beta glucan through dectin 1, and it will communicate that information down to NF-kappa B. And it could be through various channels. There's multiple channels that lead down to these different types of transcription factors. And the transcription factor goes into the nucleus of the cell, and then it induces a particular part of the genome. It gets transcribed to make some type of protein, okay, some type of chemical. And so, for example, here we can see that candida gets recognized by certain types of cellular receptors. It sends a transduction signal within the cell that activates NF-kappa B as a transcription factor. NF-kappa B moves into the nucleus of the cell, connects with an aspected part of the genome to induce interleukin 6 and tumor necrosis factor alpha. Interleukin 6, tumor necrosis factor alpha will signal neutrophils and macrophages, which are part of our innate immune system, to come in for the attack. So there's this constant loop effect that can be occurring with these chemical reactions based on the invasive nature of a particular organism like candida. Now, unfortunately, there can be negative downstream effects. Okay, tumor necrosis factor alpha has pro-inflammatory effects. So if that signal never gets turned off, now we can get stuck into a pro-inflammatory state. So just to recap, the fundamental components of the immune system are physical barriers, our innate immune system, and our adaptive immune system. These PAMPs, these pathogen-associated molecular patterns, are these molecular configurations that are unique to a specific pathogen that get recognized by our innate immune system pattern recognition receptors. Okay, so those things come together in this unique relationship because it's the PRRs of our body that are recognizing these different molecular patterns. But remember, there's only so many of these PRRs that exist. Okay, and this is why the innate immune system is not enough to have full-blown, adequate, healthy immunity. We know that candida is an opportunistic organism, and so it can take advantage and actually invade the mucosal tissue within the digestive tract. But because candida has its own unique PAMPs, it will get recognized by innate immune cells that are carrying specific PRRs against it. That beta-1,3-glucan PAMP is something that is highly recognizable by our pattern recognition receptors. So candida will try and hide it as best as possible. But it eventually gets recognized through various other means. And when it is recognized, there is a transduction signal sent into the cell that activates NF-kappa B, for example, as a transcription factor, which moves into the nucleus of the cell, interacts with specific genes to transcribe that information into protein production or chemical production like interleukin 6 or tumor necrosis factor. So this has a pro-inflammatory cytokine effect. But these signals that come through this transduction transcription factor reaction will communicate back to the innate immune cells. So it'll trigger neutrophils and macrophages and other cells of the innate immune system to come into that tissue area to ramp up immune defense. And where we get stuck, or where our patients can get stuck from a inflammatory standpoint from something like candida, is when we start to get robust activity or infiltration of neutrophils. So a neutrophil is also called a polymorphonuclear leukocyte. It has multiple lobes as part of its nucleus. That's how it's, for the most part, is identified. And it's part of a grouping of cells called granulocytes. So that would be, granulocytes would be neutrophils, basophils, eosinophils, for example. I think it's eosinophils. Yeah, or it might be mast cells. Anyway, so they contain, we know that neutrophils contain these granules, which I just showed you previously. So let's imagine that we've got candida that is invading into a tissue space. So just imagine this is within the lining of the digestive tract, for example. Now, within our tissue spaces, we typically have what are called resident macrophages. Macrophages are monocytes, or were monocytes, that transformed into a macrophage and then took up shop. So the main resident innate immune cells we have in our tissue spaces are macrophages and mast cells. Neutrophils, basophils, eosinophils are in general circulation. They have to be called into a tissue area to do their job. So we've got these resident macrophages that are kind of hanging out. So for example, a microglial cell is a macrophage within the brain. We have Kupffer cells, which are macrophages in the liver, alveolar cells, which are macrophages in the, or alveolar macrophages are in the lungs, for example. Okay, so we've got this tissue space that's now being invaded by candida. Well, there are chemical signals that are sent that trigger a circulating neutrophil to stop in its circulation. So this guy will come over, kind of get stuck to the endothelial lining, for example, and then it'll start to move into that tissue space. So this PMN, just think of it as a neutrophil. Okay, so here's a pathogen, for example, this little red oval shaped thing. So the neutrophil will come in and it'll attack it. Like, let's say that candida organism, which we saw before. So it'll engulf it. And once it's engulfed that organism, it becomes what's called an apoptotic PMN, or an apoptotic neutrophil. And it kind of hangs out. It needs assistance to eventually this entire thing to get phagocytized by a resident macrophage. So of course, there are chemical signals that are sent to a macrophage to come over and engulf that apoptotic neutrophil and then remove it through the draining lymphatics. Okay, so we initially have neutrophil infiltration, engages the pathogen, signals are sent to a macrophage to come in, swallow the whole thing up, turn off the signal, get rid of the debris out of the draining lymphatics, and calm down the tissue from inflammation. So that's how it should work. Well, there could be scenarios where we have a failure of resolution. Perhaps we have so much tissue invasion that the neutrophils themselves become overwhelmed, or the macrophages become overwhelmed. There are actual substances that are produced sometimes because of these reactions that actually inhibit macrophage activity. So if we have a situation where a neutrophil has engulfed a pathogen, but it can't adequately trigger a macrophage, or there aren't enough macrophages, or the macrophages aren't responsive, there's a limited amount of time that these apoptotic neutrophils have before they start to degranulate. It's around 10 hours or so on average. And if these things don't get cleared from that tissue space, they start to degranulate. They start to break down and start to release their internal compounds. Well, these compounds are enzymes, and these enzymes would then cause local tissue destruction, which themselves drives further inflammation. When we have a cell that has a mild injury, or a cell, for example, that, let's say, is in a process of dying, there are internal signals within our cells called DAMPs, danger associated molecular patterns. And when those things get expressed, there is a chemical reaction that will trigger an apoptotic reaction within that cell. So apoptosis is this cell suicide death response. And a macrophage will come in, or a neutrophil macrophage interaction will come in, clear out that dead and dying cell and remove it before the internal contents of the cell start to be exposed to the immune system. And so that's a normal reaction. The immune system essentially stays quiet. But when we have a severe injury, or we have overactivation of inflammation in a tissue space, those DAMPs that would normally be contained within the cell all of a sudden start to spill out. Okay, they start to become recognizable outside the cell confines, and they themselves start to be recognized by the immune system. In severe cases, this can lead to necrosis, for example. They themselves get recognized by certain types of pattern recognition receptors, and they will elicit an immune response, and they can elicit a chronic inflammatory immune response. The problem is if that response is very significant, because these are molecular structures of our own body, they can now elicit an antibody or immune system response against our own tissue. And this is one of the dangerous things about prolonged inflammation in the digestive tract from dysbiosis or invading organisms, is the potential to cause epithelial destruction and the release of DAMPs that would then trigger immune response that cause cross antibody reaction to other areas in the brain or other areas in the body. Okay, so we know that these PMNs, these neutrophils, will enter tissue areas to engage a pathogen. Once they've engaged it, they turn into an apoptotic neutrophil or apoptotic PMN. They're signals that are sent to resident macrophages to come over, gobble the whole thing up, and remove it from the draining lymphatics. But if the system is overwhelmed, or we're not getting adequate activation of macrophages, then we can't clear those apoptotic neutrophils. After a period of time, they're sitting in the tissue space, they break down, cause more tissue damage, more inflammation, and cross signaling with regards to autoimmunity. Now, I've used this slide quite a bit in many of my courses and other webinars that I've done. So we've got candida that can exist as a individual organism. And for the most part, if our microbiome is well adapted, diverse, it's going to keep things in check for the most part. Okay, so pretty much everybody's happy. And so here's a dendritic cell, by the way, sticking out one of its arms just to sample what's happening and passing through the lumen of the digestive tract, for example. But there's many things that can tip the scale for candida to become more invasive and pathogenic. So it forms different proteins that allow it to become endocytosed, or for the hypha to become invasive. If you grow a hypha that goes right through an epithelial cell, it will engage a PRR. Okay, well, that engagement will be triggering different transcription factor or different chemicals that get produced through transcription factor activation. Interleukin 6 we mentioned before. We could add NF-kappa B in this picture. We can also generate things called inflammasomes. And inflammasomes are these highly complex structures inside these immune cells that are related to inflammation as well. Okay, so a number of different inflammatory or immune activating cytokines can be triggered. Interestingly, we can trigger something called Th17. Th17 is part of the T helper cell system. Th17 in part is responsible for surveying the mucosal layers of the body. So just think of the lungs or the digestive tract, for example. And Th17 produces a cytokine called interleukin 17 that triggers a neutrophil to attack a hypha. So here we have part of the adaptive immune system coming and communicating back to part of the innate immune system to deal with this invading organism. Interleukin 22 gets produced. Interleukin 22 produces or stimulates the production of defensin peptides. These little defensin peptides act like little hand grenades that get thrown out to try to attack an invading candida. So many other mechanisms are involved in this scenario. So I'm not going to get lost into this because it can be complicated, but I just want to point out a few things. I mentioned before about MyD88, okay, this transduction reaction that occurs. We know that MyD88 is connected to certain types of toll-like receptors, which is a type of PRR, a type of pattern recognition receptor. And MyD88 is connected to NF-kappa B. It's not the only thing connected to NF-kappa B. We can get NF-kappa B activation through other PRR transduction reactions. Okay, but we know that when NF-kappa B is triggered, it moves into the nucleus of the cell, interacts with the genome to transcribe the production of different immune chemicals like interleukin 6, for example, or tumor necrosis factor alpha. So all of that we've kind of covered. And that is a normal response, right? That essentially is a normal process of immune engagement. And we know that certain cytokines that get produced can activate aspects of our adaptive immune system. Well, for example, these chemicals can trigger through different receptors. These are T cell receptors sitting on Th17 CD4 helper cells. Okay, and I just mentioned that Th17 is one of the things that it does and is involved in is helping to provide immune activity along the mucosal surfaces of our body. So it's going to either produce other cytokines that induce production of defensin, for example, or trigger neutrophils to attack a hypha. Okay, so that's sort of typical, a normal reaction. Well, as you can imagine, there's different layers and complexity to the immune system. And so, again, I mentioned before that the type of antigen that we're coming in contact with and its molecular configuration and where it goes when it's incorporated into a dendritic cell and how it's processed will determine in what kind of information is passed on to a naive T cell. And therefore, it can in part direct, are we moving towards a Th1, a Th2 type of side of the equation? Well, notice that Th17 is involved in dealing with extracellular bacteria. It's involved in dealing with fungi, for example. It's not going after intracellular pathogens. That would be more of a Th1 response. Okay, it's not really going after parasites to a significant degree. That's more of a Th2 response. Th17 is really geared towards these extracellular bacteria and fungi. Now, fungi, if you remember, is yeast, which could be candida. It could also be mold. So Aspergillus penicillium, for example, would be examples of fungi. Also notice we have what are called Tregs, which are T regulatory cells. And T regulatory cells are important for overall immune regulation and immune tolerance. Well, if we have a prolonged, overexpressed Th17 response because of prolonged dysbiosis, candida, fungi existing in the digestive tract, etc., we're potentially driving up Th17 reactions. And when this system is overdriven, it can inhibit T regulatory cells. which means it can decrease immune tolerance. It can decrease lymphocyte homeostasis. It can decrease regulation of the immune response. And this Th2 response plays a big role in autoimmune reactions as well. Well, it turns out that irregulatory cells have a regulatory role over Th17. They have a regulatory role over Th1, Th2 as well, but these two systems within the T cell scenario are really kind of intertwined. And so it's just from a basic standpoint, kind of understanding that. So what am I getting at? Is that candida in an invasive form has the ability to generate many different types of pathogenicity mechanisms, but it can be linked to autoimmune reactions and cause a downregulation of T regulatory cells, which totally disrupts immune cell regulation. So to recap, mucosal invasive candida triggers various cellular responses. So it can trigger that defensin peptides, those little hand grenades that get thrown out. It can trigger inflammasome activation. It can also induce Th17 cells, and that's where we get the interleukin 17 produced. And all of that's fine in a regulated fashion. But too much invasion, breakdown of the lining of the digestive tract, okay, all of a sudden starts to overwhelm these systems. And that's where we get stuck into a process of chronic inflammation. Okay, and so T helper cells have a suppressive effect to some degree on T regulatory cells. T regulatory cells will increase immune tolerance. So if we have way overexpression of Th17, we're now losing immune tolerance. And another thing to think about in this is that immune tolerance also means immune tolerance to many of the substances that we come in contact with, particularly foods. And so whenever you think about candida or you think about the microbiome, my recommendation, this is what I do anymore, is I don't just get locked in to thinking that it's just a single problem. This whole notion of how do I kill the candida, right? These are very complicated, intertwined organisms that are part of the entire ecosystem of our body. And so other things that we come in contact with, solvents, pesticides, herbicides, metals, okay, this information actually comes from that article that Dr. Vajani from Immunoscience wrote a number of years ago. You can actually get it online for free. There's a part 1 and a part 2, by the way. Because as you can see, if we're coming in contact with other environmental factors, that they themselves are causing tissue damage. The tissue damage can lead to self-antigen presentation. These danger-associated molecular patterns are molecular patterns internal to our body. And if they get presented to the immune system, they themselves trigger their own immune reaction. They themselves create inflammation. And they themselves then can be an inducing factor for different types of autoimmune diseases. So it's a collection of things that we're exposed to. Okay, and so much of what we do as integrative and functional medicine practitioners is trying to figure out what are the factors that might be inducing poor health in somebody. But fundamentally, when we get down to it, it's really trying to improve the health and functionality of the digestive tract as much as possible. And this is why when you look at individuals who have chronic infections, dysbiosis, bacterial problems, fungal problems, mold problems, mycotoxin issues, they're not typically just reacting to one thing. They can be reacting to multiple things that are in their environment, things that they're ingesting, and all of these things are, I shouldn't say all, in this particular case, we can see a lot of things lighting up. This actually comes from Cyrex Lab, their multiple mucosal immune reactivity screen. This person's highly reactive to lipopolysaccharides, which are part of gram-negative bacteria. They've got leaky gut. They've got immune reactions to their claudin and zonulin, for example. They've got high reactivity to gliadin proteins. They've got some reactions to things like corn or eggs, okay, and other environmental compounds, mercury, chemicals, and they're even now cross-reacting to different neurological tissue like the blood-brain barrier. So we can not only develop a leaky gut, we can develop a leaky blood-brain barrier, which is now leads more into the potential problems of neurodegenerative and neuroinflammatory conditions. Where does Clostridia get mixed into this whole scenario? If you've listened to some of my webinars or perhaps taken part of some of the courses through Integrative Medicine Academy, or maybe you've taken my organic acid test seminar from Great Plains, which is now Mosaic Diagnostics, you've probably seen a few of these slides. Clostridia is another sort of very sophisticated organism that we can come in contact with, and we're all more prone to it when our microbiome is greatly disrupted. It can cause, at least in the C. difficile form, serious infection, and many deaths have occurred every year in the United States from C. difficile infections completely overwhelming somebody with regards to inflammation, bowel toxicity, and immune dysregulation. So the two primary virulent factors of inflammatory bowel disease for C. diff is these toxins A and B. And these were discovered many years ago and associated with a condition called pseudomembranous colitis. And so this leads to leaky gut, inflammation, vascular swelling. Well, these things are toxins, okay? Well, those toxins have their own molecular pattern. And the inflammation that occurs in certain people, if it's so severe and so serious, it can actually be lethal. Most of the testing that is done to assess for Clostridia infections is primarily just focused on Clostridia difficile. The last article I read, I can't remember the date now, but it mentioned there's about 36, I think, different strains of Clostridia difficile. So there's not just one, there's multiple strains of just this type of Clostridia. Now, not all of them produce toxin A and B. The toxin A and B are the main virulent factors within the digestive tract. But if you've done organic acid testing, you know that the organic acid test will pick up on other compounds that get produced by Clostridia bacteria, including other strains of Clostridia difficile. So for example, this is just somebody who had high levels of toxin A and B. This was picked up on a Diagnostic Solutions laboratory stool test. Doctors Data has their own comprehensive Clostridium culture. So in this particular person, you can see a 1+ Clostridium difficile. This isn't looking at the A and B toxin, by the way. This is just identifying different types of Clostridia bacteria. Well, the organic acid test gives us insight as well. And so if you're used to seeing this test from Mosaic Diagnostics, 15, 16, and 17 are the most common Clostridia markers showing up on the oat. For creosol is very specific to Clostridia difficile. But remember, there's different strains of Clostridia difficile. They're not all producing the virulence factors A and B that are damaging the gut. In fact, creosol has more of a negative effect within the brain and nervous system. The HPHPA is another type of Clostridia toxin from various types of Clostridia difficile, but not C. diff. Okay, so these compounds tend to have an adverse effect in the brain and nervous system. And what they do is they primarily affect the function of dopamine beta hydroxylase, which converts dopamine to norepinephrine. Okay, and so when you inhibit that particular enzyme, you're going to affect dopamine metabolism, and that can have a downstream effects with regards to poor energy production, for example, or too much dopamine. Too much dopamine in the brain and nervous system can be toxic. So it has its own negative chemical influence. And you'll have plenty of people who will have presence of organic acid test, even the 4-creosol, but no evidence of any bowel disease. So we can't just rely on bowel symptoms as an indicator of, of Clostridia problems. So a very interesting article, this came out in 2013, called The Host Immune Response to Clostridia difficile Infections. Now, they're specifically looking at the type of C. difficile causing bowel disease. Okay, so they're primarily looking at things that would be inducing the production of or producing virulence factors A and B. But I want you to look at this in the context of what we've just discussed. So they're mentioning here that intestinal homeostasis is regulated by a ratio between Th17 and regulatory T cells, which I just mentioned before, that when we have mucosal infections, candida being one of them, we can tip the scale the other way, where we have too high of a Th17 and too low of a T regulatory cell. So things are kept in check, balanced, when we've got low level of dysbiosis, okay, going on, and or optimal as much as possible microbiome diversity. There's always going to be some sampling that occurs by those dendritic cells. This is a dendritic cell right here that is sampling things moving through the gut. Okay, but if things are well balanced, we're getting proper signaling and balance between the regulatory and the Th17 cells. Now, we all know that many people can be induced into having a C. difficile bowel infection by being on antibiotics. Okay, so antibiotics come in and they have this broad sweeping effect on the microbiome. And all of a sudden, we get dysbiosis, opportunistic pathogens that might be present, now seize the opportunity. And now all of a sudden, we're getting an increase levels of Th17 and a suppressive effect on T regulatory cells. Well, if we've got C. difficile sitting there, okay, it's now going to, because it has its own intrinsic desire to thrive and survive, it's now going to proliferate, and it's going to start spewing its toxins. And those toxins are going to have a damaging effect on the mucosal lining of the digestive tract. When you start damaging the lining of the digestive tract, the innate immune system tries to come in and fight it. And so it starts coming in, the monocytes coming in, the neutrophils start coming in, and we start getting neutrophil activation. Okay, and all of this can lead to inflammation within the digestive tract. In fact, pseudomembranous colitis is this hugely profound inflammatory reaction occurring because we have the innate immune system that's trying to battle back these toxic bacteria. And then, of course, we get antigen presentation crossing over to the adaptive immune system a few days later, and then it too will get into the fight. And when you actually study this from a therapeutic standpoint, there's all kinds of therapeutic trials out there of trying to come up with ways to block the binding of C. difficile on the mucosal lining, to try to calm down neutrophil activation, to try to even come up with monoclonal antibodies that could bind themselves to these organisms or affect other aspects of the adaptive immune system. So the same principle applies with regards to invasive candida that can be applied to the manifestations of Clostridia. But the way I look at these types of things and what I've seen over the years in clinical practice is that people who generally have chronic dysbiosis because of poor microbiome status, who have chronic candida issues, often have more problems with Clostridia, and the more problems they have with Clostridia, the more problems they have with candida. And so again, it all starts to get intertwined. And then, of course, on top of that, if these situations are chronic over months, if not years, we can start to have, well, not even, in many cases, it doesn't even take that long. You then apply on top of that environmental exposures, food reactions of various forms, And we can see why so many people are locked in a state of chronic dysfunction, chronic inflammation, chronic immune dysregulation. Because ultimately, as we start to lose functionality of the lining of the digestive tract, whether that's because of lack of biodiversity in the microbiome, the lack of adequate mucin production to maintain a thick mucal layer, the suppression of secretory IgA, which would also get intertwined with all of this, damage along the mucosal lining because of invading pathogens like candida, you're getting more and more cross presentation between these antigen presenting cells that are presenting that information to the adaptive immune system. We're producing a wide variety of antibodies. And if we start damaging our tissue and we start expressing these internal molecular patterns of our own body, well, all of a sudden we're setting ourselves up for autoimmune disease, because that ultimately is what then can trigger an immune response to our myelin, to our blood-brain barrier, to our joints, to our actin, to components of our mitochondria, to our heart, wherever it may be. So this is how people a lot of times end up with things like MS and rheumatoid arthritis and lupus. So what are some strategies? Well, you know, that is in itself is a lengthy discussion, but much of it comes back to trying to restore some normalcy of our microbiome. Okay, getting rid of reactive foods, incorporating more plant-based foods, fiber, multi-strain probiotics, etc. Okay, the more we can improve the microbiome status, the more we can sort of regulate our immune response. Yes, there's sometimes we need to deal with the pathogens like Clostridia, other bacterial infections, parasitic infections. Many of our patients are exposed to mold and mycotoxins. None of us live in a bubble, so there's always going to be the potential for environmental chemicals. Glyphosate, for example, we know can have a damaging effect within the microbiome. So it's not typically just one thing. It's not like you can just sit there and throw a nice statin at somebody or give them a botanical and call it a day. Okay. In most people who are dealing with chronic issues, it's much more complex than that. Their problem didn't develop just over a few weeks. Okay. And so the lifestyle factors, the dietary factors are very much part of this. And this information and more we get into in great detail in our Candida Mastery Course through our Integrative Medicine Academy. We actually have a new course starting November 13th. And so if you're interested in this information, you're interested in learning more about the immune reaction to Candida, how things are recognized, how things are sequenced, and diving into this information in much more detail, I would encourage you to go to candidamasterycourse.com and check out the information of the course. If you have any questions, you can always email us at candidamasterycourse@gmail.com. Now, I told you at the beginning of this lecture, I'd recommend everybody kind of hold on because I wanted to present some new information. And you can actually find this article. This is a hypothesis article from Dr. Shaw. Dr. Shaw was the original founder of Great Plains Laboratory. Great Plains Laboratory was now became Mosaic Diagnostics, but Dr. Shaw is still actively involved in research. And so he released this article this past year called Inhibition of Beta Oxidation Pathway of Fatty Acids and Dopamine Beta Hydroxylase. Now, this was specifically geared in the discussion of autism. But what I would encourage you to do when you read this article is don't just focus on the autism aspect, although that's an important aspect of this, but think and extrapolate the information of how it might affect somebody else. So we know that the organic acid test from Mosaic Diagnostics primarily detects HP-HbA about 80% of the time, 4-creosols about 15% of the time, and then 4-hydroxyphenylacetic comes in at about third position. And we know that these toxins adversely affect the dopamine beta hydroxylase. gets pretty heavy pretty quick into some biochemistry. And so I'm not going to spend a tremendous amount of time on this, but the point is this, is that there are various metabolites of these Clostridia bacteria. So when the body is, or Clostridia in the digestive tract is producing this HPHPA, it gets metabolized through our own fatty acid pathways and our own biochemistry. And in order to properly metabolize it, we need good nutrition. So we actually need glycine, for example. And we also need coenzyme A. Now, coenzyme A is linked to B5 metabolism. Free coenzyme A is involved in many different biochemical reactions. So what Shah is pointing out, okay, is that in the presence of C. difficile, okay, producing 4-hydroxyphenylacetic or other Clostridia producing HPHPA, we know that those things can inhibit dopamine beta hydroxylase. As we inhibit dopamine beta hydroxylase, that can lead to an increase of dopamine, which has its own toxic effects within the brain and nervous system. Various Clostridia bacteria also tend to produce other types of compounds, and they produce something called phenylpropionic acid. And I'll talk more about these things later through, whether it's through other videos or webinars or some of the courses that we have. But phenylpropionic acid gets metabolized into something called 3,3-hydroxyphenylpropionic acid, which is essentially HPHPA. Well, in order to process this normally, we need coenzyme A. Well, coenzyme A gets linked to the metabolism. We've got to somehow get rid of this. So we link it to coenzyme A metabolism. The problem is as we suck up more coenzyme A, we leave less for downstream effects in other areas biochemically. So this affects fatty acid metabolism, or can affect fatty acid metabolism in other areas. Fatty acid metabolism is quite significant for mitochondrial activation. We can get decreased cholesterol production. As I mentioned, we get decreased free coenzyme A activation. Well, the problem with that is that coenzyme A is involved in many other types of enzyme reactions linked to body metabolism. And what Shah ended up coming towards, and I've actually looked into this in more detail, and it's very, very complicated, but by having inadequate coenzyme A, free coenzyme A, because of too much HPHPA coming from certain types of Clostridia bacteria, which, by the way, can become more problematic the more our microbiome is disturbed, the more chemicals we're exposed to, like glyphosate, and potentially the more problematic it can be if we have also going on immune dysregulation linked to chronic candida. In that a lack of free coenzyme A leads to a decrease of something called sonic hedgehog. And sonic hedgehog is a protein that is critically needed for proper brain development. What happens is in kids, basically in developing embryos, if they don't get activation of their sonic hedgehog, they have all kinds of neurological deficits. They have digit formation problems. But sonic hedgehog is a protein that is produced throughout our life. And it's necessary to maintain proper neuronal signaling and structural configuration. And so this is really important, profound information, very much applicable to the autism community for sure. So, but I think it's also quite significant to other types of patient populations. So it's another complicating factor with regards to Clostridia. And this is why it's so important, again, not to hyper focus on Clostridia as only having gut problems. So in January 2024, we're releasing a new course. So I mentioned we have a new version of our Candida Mastery Course coming up later this month. In January 2024, we're launching a new course on Clostridia. And so what I'm going to be doing is looking at the information with regards to Clostridia, and then as we move that over and start looking at the influences and other aspects of Clostridia bacteria. So I'm really excited about this course we have in development coming up in early 2024. So just pay attention to that. We'll be making some announcements on that later. There are some other areas where you can follow me. I do have a Substack. You can go to Dr. Woeller at substack.com. I look to post things on my Substack at least twice a week. It's all free information. It could be articles. I might be sharing information from other people I'm following, videos, for example. So Dr. Woeller at substack.com. Another cool area we have is on YouTube. So if you actually go, you just type in Integrative Medicine Academy on YouTube. We have our own channel. And this is a slightly older slide, but we have a lot of past videos, and I've started creating what are called mechanisms videos. And so again, this is all free information. And the mechanisms videos are very interesting. They're usually anywhere about 6 to 8 minutes in length, sometimes maybe a little bit longer depending on the topic. But I'll pick a specific topic that related to functional integrated medicine, things that I'm learning, that I've taught about, or might be part of the courses that we have, and I'll go through them. So this example here on mitochondrial melatonin, on SOD activation, for example, is very interesting. I'm creating a whole series of short videos on different pathogenicity mechanisms of candida, for example. So there's an endless amount of topics. So again, if you're interested in that, you can go to Integrative Medicine Academy on YouTube. You can subscribe to it or just view it. So a lot of fun, a lot of cool information. And then if you're interested in just general information about Integrative Medicine Academy, go to integrativemedicineacademy.com. You can look at our full list of courses, our mastery courses that we have. And then if you have any questions, please reach out to us at integrativemedicineacademy@gmail.com. Well, thanks so much, everybody, for your attention. I really appreciate it. I hope you found this information interesting. I know some of it might be new information to you, but it's all important. And these are things that I'm researching and connecting to and applying in my own clinical practice. So it's important I think for all of us just to keep digging, asking questions, trying to find information that makes sense and how these things apply to patients with chronic illness. So that's what we do. That's what I do through Integrative Medicine Academy and other channels that I'm involved in. So I appreciate all of you. Again, I'm Dr. Kurt Woeller for Integrative Medicine Academy. Take care. Thank you.